I'd like to introduce our first speaker, Tony Kouzarides, who is a professor at Cambridge University. He is a Royal Society professor and also deputy director at the Gurdon Institute. Tony did his PhD in Cambridge and his postdoctoral work at the LMB, where in fact we not only overlapped, but we shared for a short period a bench. And I have to say I was quite pleased when Tony swaned off to New York for bigger fishes to fry, because I could take over the bench that Tony had, the better end of the bench that we were sharing. In New York, he worked on functional aspects of C-FOS, and when he returned, he joined the Gurdon Institute, where he has been ever since then. And this is really where he had made a lot of discoveries that you will know about in the field of chromatin and how these changes relate to cancer. And I'm sure many of these aspects will be highlighted and touched on in his keynote lecture. So while I think many of you will know Tony from his outstanding work on chromatin, fewer people will probably know that he's also very business savvy. So Tony has founded 2 companies. The first company is Chroma Therapeutics, and the second will be familiar to many of you, and it's Upcome. And it shows that when Tony, probably when he returned from the US, he found he needed antibodies. The antibodies weren't there. So what do? You either moan about it or you set up a company that makes the antibodies. So he set up a company that makes a lot of antibodies that, of course, many of you will have used. In addition to, I think, an increasing collection of medals and prizes, Tony has been honored and his work in many different ways. He is a member of EMBO, he is a member of the British Academy of Medical Science and the Royal Society. So we are absolutely thrilled today that Tony will be giving the first of our keynote lectures, and he will be talking about chromatin modifications and their role in cancer. And it's really important that we take on board the many discoveries that have been made in cancer and then see how these discoveries really can be utilized in the common diseases that many of you in the audience here will be working on. So many of the discoveries will be made in cancer first and developed before they then will be applied in common diseases. So that's all I wanted to say to introduce Tony. So Tony, thank you very much for joining us today.
Well, thank you, Stefan, for that introduction. Thank you for all the things you said. And thank you for organizing this meeting in my hometown, which is nice to just come here by bicycle. Cambridge is a great place. It's a great city. You know, you have a great life, lovely to work in. And those of you who are interested in coming to Cambridge to work, I'd like to say that the weather here is always brilliant, like it is today. Even on a slide that I have, the weather is brilliant. So it really never rains in Cambridge, so forget about Barcelona, just come to Cambridge. And now for my talk, which will have a lot more truth in it. So I'm going to talk about epigenetic modifications. We're interested mostly in histone modifications, of which there are many. A lot of these pathways we know nothing about. We know the modifications exist, we don't know what they do. We're interested in understanding the biological pathways that they regulate, the mechanisms by which they regulate them, and their implications specifically in cancer. And we know that there are many connections to cancer already. This is the list of genetic damage in genes from the Broad and the Sanger Center. And you can see that there's damage in enzymes that put on the modifications, enzymes that take them off, the proteins that read the modifications, the chromatin remodeling activities they recruit, and even in the histones themselves. Point mutations in residues in histones suggest that these are direct pathways that regulate histones. And therefore, these pathways are good targets for drug discovery and potential cancer therapy, which we are interested in. So we became interested in this field many years ago when Andy Bannister in the lab identified a coactivator, CBP, as a histone acetyltransferase. Now, this was the same year that the first enzymes that modify chromatin, acetyltransferases, were discovered. So Dave Alice discovered GCN5, and Pat Nakatani discovered PCAF. And these were the first identities of enzymes that could put on modifications on histones. Now, at this early time point, we became aware that these pathways may be implicated in cancer because what Andy did to identify this as an enzyme was to use the E1A oncoprotein as a bait. And we knew that the residues that bound CBP and the relative P300 are involved in oncogenesis, and therefore we made the leap that this protein, and therefore now this enzyme, may be involved in cancer. And therefore, soon after, we collaborated with Carlos Caldas' lab to look for mutations in cancer in these genes, CBP and P300. And indeed, we find that many cancers carried mutations. And this was essentially the first genetic evidence that there are damages in these pathways that mediate modifications of chromatin. So these two discoveries then led us into two different paths at the time, which are still going on at the moment in the lab. The first is to identify small molecule inhibitors to disrupt pathways in cancer, and the second is to identify new modification pathways of chromatin, other than acetylation, because these may also represent new targets for drug discovery. So I'm going to briefly mention these two programs in the lab, and then tell you about two different stories that we have, which are unpublished and still going on. So our foray into drug discovery came early. We collaborated with Mirla Hearn in a CI, and we looked for inhibitors for CBP and P300, set up assays, discovered them, but these were not very potent. So essentially, they didn't go very far. We knew you could inhibit the activity of these enzymes, but they weren't really good inhibitors. Paul Marks, on the other hand, was able to change his SAHA HDAC inhibitor into vorinostat, which is a drug that was approved for cutaneous T-cell lymphoma. So we knew that these inhibitors were actually safe in man and potentially useful. So this was quite early. And now, the new class of proteins that are being targeted are proteins that recognize the acetylated lysine. These proteins have got a small pocket that recognizes the modification, and therefore they make a good substrate for drug discovery. So our contribution to this came when we collaborated with GSK. They were developing an inhibitor called IBET, which disrupt the binding of the BET proteins to acetylated lysines. They were developing this for immunoinflammation. We recognized that there was a potential for using them for leukemia. We asked for the inhibitor, and then we were able to show that the BET inhibitors could be used against MLL leukemia in both cell lines and in mice. And based on this discovery, GSK has now set up trials in Cambridge for AML leukemia, of which MLL is one. And these are being carried out by our collaborators, such as Brian Huntley in Cambridge. So we await the results of those trials eagerly. And now we're looking at a number of different inhibitors from companies that are giving us the small molecule inhibitors against epigenetic targets to see if there's another potential pathway that could be disrupted in this way. So hopefully more to come. Now, the other program was the discovery of new modification pathways. So following the discovery of acetylation, modifying acetylation enzymes, there was a discovery by Mike Scottcomb's lab of arginine methyltransferases, and then Thomas Yenuan discovered lysine methyltransferases. So these are quite common modifications. Then we started ourselves looking for other modification pathways that might have been missed. And over the last 10 years, we've described citrullination, conversion of arginine to citrulline, protein isomerization, tyrosine phosphorylation, and most recently, a few months ago, methylation of glutamines. Now, all of these modification pathways are involved in transcription, and all of them are connected to cancer in some way or another, some more than others. Mechanistically, we know that these pathways work by recruiting proteins or disrupting proteins or talking to each other. So acetylation recruits bromodomain proteins, arginine methylation pseudodomain proteins, lysine methylation chromodomain proteins. Citrulination disrupts the binding of H1. Proline isomerization is a non-covalent modification, so it affects the modification next to it, lysine methylation in this case. Tyrosine phosphorylation disrupts AP1 binding, and glutamine methylation prevents FACT binding. So these are very common mechanisms that appear to be mediated by these pathways. Now, the first three modifications are fairly common, and they could potentially affect any pathway that leads to DNA events, so replication, repair, transcription. These pathways that we've been discovering may have more esoteric, not esoteric, more dedicated functions. And this is something that we didn't know about until this year, because our work on glutamate methylation and also citrulination of arginines shows that these modifications may be dedicated. And I'll tell you what I mean in the next few slides. So glutamate methylation is mediated by fibrillarin in human cells and NOP1 in yeast. It methylates a single glutamine on histone H2A, and it affects only ribosomal DNA transcription. And this dedicated function comes from its localization, because this enzyme is localized only in the nucleolus. So this is an antibody that recognizes the modification. This is an antibody that recognizes the enzyme, and they're both colocalized only in the nucleolus in mammalian cells. If we now look in yeast and map where this modification lies using the antibody, ChIP-seq analysis, there is only a single peak on the entire yeast genome. This peak lies over the ribosomal DNA locus, which is where this modification has a function. Again, very dedicated function because of its localization. So glutamate methylation is the only modification discovered so far that has a dedication to a single polymerase, POL1, because this is present in the nucleolus and regulates ribosomal DNA transcription. If we now go to citrulination, and the enzymes that modify this is PADI4. This has got dedication because it's present only in certain cell types. So 10 years ago, we knew that this enzyme was only expressed in neutrophils, and we showed there that it has a function to modify histones and affect transcription. But recently, Maria Christoforou has found that this enzyme is also expressed in pluripotent stem cells. So two different cell types in which you find this enzyme. And we think that you find this in two different cell types because the function is very dedicated to those cell types. So if you change neural stem cells, reprogram them to iPS cells, the enzyme is expressed, and then it mediates decondensation, which is necessary for iPS cell function. In neutrophils, you have bacterial infection expressing PADI4. This is then mediating decondensation in killer cells to allow chromatin to be thrown out into nets in the medium to capture bacterial components. So these two cell types have a necessity for decondensation, and this enzyme comes on to mediate that function. And I'd like to point out that in cancer, PADI4 is almost always comes on when you have more metastatic cancer. In benign cancers, it is off. But in metastatic cancers, it comes on, probably because you need this decondensation function generally for cancer cells. So that's the background. What I'm going to do now is tell you two stories, which, as I said, both are ongoing and both are unpublished. One is about a new modification pathway of histones, and the other is on non-coding RNA in transcription and cancer. So the new modification I'd like to describe is a lysine methylation, which has a role in DNA replication. And this is the work of Elena Santos-Rosa, primarily with help from Peter Tezans. So what we know about DNA replication and modifications is nothing. We know that these two modifications, K36 monomethylation and K37 monomethylation of H3, are enriched at sites of DNA replication. That's all we know from this patient from Toshi Tsukiyama using mass spec analysis. Now, what happens at DNA replication sites? Well, simplistically, you have the origin recognition complex bringing in the MCM helicase, which then is required for DNA replication. This is a very simplistic view. So in order to work out what these modifications do in this system, we raised an antibody against monomethyl 37, which didn't exist, and used an already existing antibody against monomethyl 36. So what do these modifications look like on replication origins? Well, what we see is that these two modifications are dynamically changing. So here you have yeast cells that are blocked and then released into S phase. And you now look at time points into S phase. You can see that monomethylation of 37 goes down and then comes up. 36 monomethylation goes up and then comes down. So at 10 minutes into S phase, they anti-correlate. Now, if you look at one of the components that I just showed you that also hangs out at replication origins, the MCM2 protein, one of the MCM hexamer, you find that that also dynamically changes, and its dynamics look like 36 monomethylation. And we think that these modifications can directly affect the MCM2 protein, because the MCM2 protein in this paper was shown to bind this region of histone H3 where these two modifications lie. Now, do these residues have any role to play in the recruitment of MCM2? The answer is yes, because if you mutate these two residues to alanine, you find that you lose the binding to MCM2. The crucial question is what happens with methylation, and now I'm going to show you what we know about the methylation pathway that leads to K37 methylation. Now, if you monomethylate this peptide, the histone peptide, what you find is that you have the same result as point mutating it to alanine. You reduce the binding to MCM2. So essentially, if you mutate the site to alanine or methylate it to monomethylated, you have the same consequence. They phenocopy each other. And therefore, we can now use this mutant in yeast in vivo to see what happens at replication origins if you methylate lysine 37. So what are the features of that mutation? Well, the first thing you see here is that this mutation shows a delay in G1 to S transition. You can see this with FACS analysis and with phosphorylation of a marker, ORC6. The second thing you see is that this mutation has a defect in the initiation of origin firing. So here we've humanized the yeast cells, and now we can look at BRDU incorporation. And if you look at 351 origins of replication, in the mutant versus the wild type, you can see that you have less origin firing initiation than in the wild type. And we think that this defect is because MCM2 is not recruited because you methylated that site. It's the equivalent of methylation, because this mutant also prevents this recruitment of MCM2 at origins, which I showed you earlier. So this modification is really, this mutation is reflecting what the monomethylation would do. And all these data lead us to believe that this monomethylation antagonizes the MCM2 binding to chromatin at origins of replication. We haven't been able to go any further because the enzyme that monomethylase 37 is not known, and we haven't found it yet. We're looking for it, but haven't found it. Now, I'm going to talk about the next modification, right next door, monomethylation of 36. We think this also has to do with MCM2 recruitment, but in a positive sense now. You can see here the MCM complex, all 6 components in one, which is from Guillermo Montoya's lab. This binds to monomethylated histones better than unmodified. There's a slight change, so we think the monomethylation can hold on to the MCM2 better. And I remind you that the dynamics of monomethylation appearance reflect that of MCM2 appearance at origins. Now, what is the enzyme that methylates 36? Well, we have a candidate, which is SET2 in yeast. This is the enzyme that mono, di, and trimethylates lysine 36. It's a very well-known enzyme in mammalian cells and in yeast, and it's involved in transcription. However, there's no evidence that this enzyme is involved in replication. There's no evidence in the literature or that we have in the lab. And therefore, we started looking for a new enzyme that would monomethylate 36, but not di and trimethylate. So there are 6 set domain proteins and DOP1 in yeast that potentially are methyltransferases, but only 3 of these have been shown to have activity, all of them against histone H3, K4, K36, and K39, in all cases trimethylation. Now, we're looking for a new enzyme, and we know that none of these, we've tried them ourselves, actually hits histones. But Helena then tried nucleosomes, and you found that this enzyme, SET4, now was able to monomethylate lysine 36 on nucleosomes only. We know it's monomethylation because mass spec only recognizes one methyl group if you add the enzyme. This is work in collaboration with Michael Nielsen. And therefore, we have now 2 enzymes, SET4 that monomethylates, SET2 that monodi and trimethylates. So the question is, does this enzyme have features to do with DNA replication? First of all, if you delete this enzyme, you can see that you can reduce the enrichment of monomethylation at sites of replication. This is a meta-analysis of replication origins. On each of the sites of replication, on each side, you have monomethylation, and this is decreased. The crucial question is, does this enzyme have a role in DNA replication? And the answer is yes. If you knock out SET4, you affect, you have a delay in S phase, which you don't see if you use SET2, the transcription enzyme. And if you now overexpress this enzyme, SET4, you find that you increase proliferation and you increase, you induce S phase, exactly what you expect from a replication enzyme. So this is an enzyme that monomethylates 36 and affects replication, and it does so at the expense of di and trimethylation as 36, because these 2 modifications are present at sites of active transcription and are mediated by SET2, a different enzyme. So now we have 2 enzymes, one that seems to affect transcription and one that seems to affect replication. Why do you need 2? Well, we suspect it's because sites of replication are actually also sites for transcription. So in order to, but we know that only one of these 2 functions can be mediated at the same time. So you probably need the 2 enzymes in order to switch from replication to transcription and back. So SET4 is monomethylating 36 and affecting DNA replication. 37 also has a role to play. They both seem to affect the MCM complex. The question is this conserved in human cells? The answer is we think so, because there is an enzyme that is closest to SET4, which is MLL5 in human cells. This has not been shown to be an enzyme, but it's been tried on histones and not nucleosomes, so we're now testing that. But in terms of its biological features, it has the same features. If you knock it down, you affect cell proliferation and cell cycle and induce cell cycle arrest. So these are the same functions that I just showed you for SET4, so we believe this is the equivalent, and we're trying to see if this is the enzyme. This mammalian enzyme is also involved in cancer, and MCM complex itself has many connections to cancer, so we think that this new pathway may also be relevant for drug discovery. So that's all I want to say about that. I'm now going to move completely away from histones. I'm sorry, but this is maybe relevant to histones, but we'll have to see. This is to do with non-coding RNAs in transcription and cancer. And this is the work primarily of Paulo Amaral and with help from Emma Viret, who are both in the audience. So far I've been talking about enzymes that put on and take off modifications of chromatin. Now, more recently, our lab has changed substrates, and now we're looking for modifications of non-coding RNAs. The reason for doing this is twofold. One is that we have over 100 modifications that modify RNA, but we know nothing about them. Secondly, as I will show you, we think that these modifications may be implicated in cancer. And we are perhaps the only lab that has this evidence, because a couple of years ago, we discovered one of the only enzymes that can modify RNA. There's only less than a handful of enzymes known. And this is called BCDin-3D. We found that BCDin-3D methylates the 5' phosphate of microRNAs and inhibits the processing by DICER. So it's got a functional consequence. But interestingly, this protein, before it was known to be an enzyme, was already a marker for metastasis. And we showed that if you introduce this enzyme into cells, you increase the metastatic capacity of these cells. So now we're talking about an enzyme that modifies RNA that could have a role in cancer. So we suspect that there's a lot of other enzymes that may modify these other sites that could also potentially be targets for cancer. And we have a program of identification of these enzymes, which I'm not going to go into today. What I am going to talk about today is the substrate. We know that microRNAs are involved in cancer, but we're now going to talk about long non-coding RNAs and their potential as cancer targets. And we know very little about these, and what I'm going to tell you is about a new type of non-coding RNA, which we call positionally conserved RNAs, pcRNAs. Now, what's the rationale for looking for these positionally conserved RNAs? Well, 2% of the genome is coding. 80%, this is an underestimate, is transcribed. And this is where non-coding RNAs come from. Now, if you compare non-coding RNAs, you can't see any sequence similarity. So you can't say this non-coding RNA is important because of conservation, because there's no conservation. However, the promoters of these non-coding RNAs are often conserved, so you can recognize those. So perhaps there could be conservation in position of the non-coding RNA, but not the sequence of it. Could that be the case? So we started looking for non-coding RNAs that are next to the same gene and conserved in human and mouse. So here's what I'm talking about. This is in human now. You have the SOX2 gene. And upstream, you have two non-coding RNAs. And the promoters are conserved. And if you now move to mouse, you have exactly the same situation. SOX2, two non-coding RNAs, promoter conserved. So can we find more of these? So we looked at the human and mouse ENCODE and GENCODE transcriptomes, which are in lots of different tissues and cell lines from human and mouse. And we identified 27,000 non-coding RNAs in human cells. Then the promoters of all of these were blasted against mouse to identify the equivalents. We identified human mouse conserved promoters that are near the same coding gene, and then only looked at the ones that were expressed in both human and mouse and were next to the same gene. So this is what we call positionally conserved RNAs. And there are 562 unique pcRNAs near 480 coding genes. So there's only 2.5% of the coding capacity is associated with pcRNAs. So it's a small proportion of genes that have these non-coding RNAs conserved next to them. And we've chosen to look at only spliced non-coding RNAs and the ones that have absolutely no open reading frame. It's a very stringent criteria. So what we find is that, as you would expect, there is in fact 6 subtypes of pcRNAs, if you like. If this is the coding gene, you have overlapping, you have downstream sense, upstream sense, antisense, downstream antisense, upstream antisense, and bidirectional using the same promoter. So 7 different types. The numbers for these are the following. Most genes have 1 or 2 of these pcRNAs. You can see here, most of them have 1, some have 2, much less have 3. The most common type is bidirectional. The second most common is overlapping. And the size is, the average is about 1,000 base pairs. Now I'm going to go through the features of these non-coding RNAs that we've discovered so far. Remember that this is an ongoing project. First question we've asked is there anything in common between the coding genes around which these non-coding RNAs are found? So Go analysis indicates that very clear enrichment of genes that regulate development, and specifically transcriptional regulators, go up or down. And most of these are DNA-binding transcription factors. So here's the enrichment, and I'm just pointing out some of these here. You have lots of HOX genes, you have pluripotency genes, SOX genes, regulators of liver differentiation. These are master regulators, and nuclear receptor signaling transcription factors. So this is just a sampling. So clearly, I would say pcRNAs are associated with coding genes that regulate development. Often these are transcription factors, DNA-binding transcription factors. Now, the next question is, what is the tissue distribution of the pcRNAs? The next to coding genes involved in differentiation and development, are they themselves tissue-specific? The answer is, of course, yes. Here we have the pcRNAs from human and mouse. And on this axis, we have the tissues in which they are in. They'll be looked at, and this is the expression pattern. Blue is high expression, and I'm just highlighting here there are a bunch of non-coding RNAs that are specifically expressed in the yellow bit here, which is heart. So very specific expression in certain cell types, these non-coding RNAs next to genes that are themselves specific in gene expression. So pcRNAs are tissue-specific. The next question is, are the RNAs co-expressed with the coding gene? If they're both in the same tissue, are they expressed at the same time? So here you can see the FOXA, this is an example, FOXA2 gene and a downstream transcript. Both of these in human cells are expressed in liver. Both of these in mouse cells are expressed in liver. Very specific tissue distribution, both of them being present at the same time. Not only that, but they're also co-induced during differentiation. So here you have a retinoic acid treatment of NT2 cells to differentiate them. On the left here, you have 3 antisense transcripts next to 3 coding genes. And you can see that the coding gene and the pcRNA are either downregulated or upregulated with differentiation, both at the same time. This is an overlapping transcript now of SOX2, again, downregulated with differentiation, both of them. This is a nuclear receptor protein with a bidirectional transcript now, both co-induced after 3 days. So pcRNAs and coding genes are co-expressed and co-induced. So then the question is, are the non-coding RNAs regulating the coding transcript? So is a direct regulation. And here are 4 examples where we have knocked down the pcRNA, the non-coding RNA, and looked at the expression of both the pcRNA and the next door coding gene RNA. So here is, the example is power 3 of 3. These are 3 different siRNAs against the non-coding RNA. And we can see that both the coding and non-coding RNA virtually disappear. So pcRNAs regulate the expression of the coding gene. The final data I'm going to show you are to do with cancer. And the reason we're looking at this here is because a number of the coding genes are actually oncogenes. And therefore, one possibility is that if you have direct regulation by the non-coding RNAs, these non-coding RNAs may also be oncogenic. So this is a meta-analysis of 60 different studies that compare normal and tumor primary samples and the expression pattern of different genes in these samples. 130 of our pcRNA probes for our pcRNAs were there, and 108 of our pcRNAs were actually different or expressed differently between normal and tumor samples. This is the data here. And you have striking downregulation in this case of every single colorectal cancer in these different studies. You have these 4 non-coding RNAs downregulated. You also see upregulation of others. So very specific changes in primary tissues. Now, this is not a causative effect. We need to prove that. And therefore, we've now started looking at cell lines and tissues that have these non-coding RNAs expressed. Here's an expression analysis of 50 pcRNAs and the coding genes using nanostring. And we're looking at the expression in cancer cells and equivalent tissues. And this is the sort of thing that we are getting. You can see here FOXA2 downstream transcript and the gene itself transcript. These are in tissues. These are in cancer cell lines. Very specific expression in the liver, in both non-coding and coding. Very specific overexpression in a liver cancer from all these cell lines, both the coding and the non-coding. Again, this doesn't show that it's directly causative. So what we're doing now is checking for the effect of the pcRNA on proliferation, invasion, and migration in these cancer cell lines to show causality. So pcRNAs are implicated in cancer. We don't know if they're causal yet. We hope so. If they are, then we will have a whole new set of regulatory pathways to identify that specifically affect non-coding RNAs. These will be different from the ones that hit the oncogenes themselves that we know of, and therefore we have new targets for drug discovery. The very final question that I'm going to address in the next 2 slides is a mechanistic one, which is how are pcRNAs and the coding genes co-regulated. I showed you a lot of the fact that both of them are present at the same time. How can this happen? So we suspected, or we suspect, that maybe a topological constraint exists that allows the expression of both of these at the same time. And therefore, we looked at chromatin connectivity maps generated by KIAPET to see, and these show you which regions of chromatin are in neighborhood of others. And this is the sort of thing that we are seeing. This is still ongoing analysis. But here's one example, which is the SOX1 gene, here. And this is the non-coding RNA that overlaps it. And these here are the sites of interaction. So this site interacts with that. This site interacts with that. So you can see that you have the promoter of the overlapping pcRNA interacting with SOX, and the 3' end of the non-coding RNA region also interacting with SOX. So this makes us suspect that these non-coding RNAs and the coding genes are in some sort of structure. That means that they're co-expressed. That's as far as I'm going to go at the moment. We're looking at the data more closely, but this may explain why you have coincidence. Now, this is the final slide, which raises all the questions, so I haven't really answered many of them. But these are the questions that are being raised by these analyses. So first one is, why are regulators of development regulated by pcRNAs? I showed you that only a small number of genes, coding genes, have these pcRNAs around them. So why is that? Second thing is, what do they do? What do these non-coding genes do to these particular coding genes that they don't need to do to the other genes? How do they function mechanistically? Do they bring in enzymatic activities? Do they interact with DNA? Do they interact with RNA? What is the interaction of the non-coding RNA with the gene and its transcript? And do these different PC subclasses have different functions? Or is it just that you really need to have an RNA around the coding gene, and that's sufficient for your function? Or is it that each of them can be induced by different things, can mediate different things, and mechanistically they may be different? That's something we don't know yet. And finally, are these pathways good for disease treatment? I mean, we're looking at cancer, but there's no reason to believe that these pathways won't be applicable to any disease, because we know that these are very important regulators that are being regulated. So in principle, this could go beyond cancer that we are looking at. So hopefully, we'll have the answers in about a decade. So these are the people who did the work. I've mentioned the people who did the work. So this is Helena and Peter and Paolo and Emma, Sam, who is a bioinformaticist, and Namshik, who is not on this slide, but is here at the meeting, I think. We have collaborators. Michael Nielsen did the bioinformatics for us. Guillermo Montoya gave us the MCM2 complex. Till Bartger gave us nucleosomes. And these are all people who did bioinformatics for us. I'd like to highlight Tommaso Leonardi from the EBI here in Cambridge. Thanks very much for your attention.
So Hangmeet Sui and I published a paper in Nature about 5 years ago showing antisense long non-coding RNAs that appear to regulate sense tumor suppressor genes. And one of the, I'm sort of curious, we had sort of a list, it's probably outdated already because it was based on annotation at the time, of such RNAs that are going through promoters. That's what we were looking at. How many of them sort of overlap with what you're seeing there. But I also wanted to ask you a mechanistic question. So it was clear in that work that that these generate heterochromatin, and you can see this in stem cells. And then once that has been established, as these cells differentiate, as the tumor is wont to do, you see these methylation changes. But we think it might be, at least for this sort of mechanism, that the methylation changes, higher order things, might be consequential to some primary chromatin factors, maybe some of the things you're talking about at the very beginning of your talk. So sort of a big question, but I apologize for that, but very curious on what you have to say.
In general, we're looking at modifications at the moment as to where they map exactly. We haven't mechanistically looked at modifications. I'm sure they are important, where we will look at them. We haven't done a lot about mechanism, and we haven't really looked at all the non-coding RNAs that actually are present in our data set that have a function of some sort. There's too many of them to look at. We're looking generically at the function. And maybe some of them will have a distinct function rather than one I'm just describing. That's what we need to find out.
So how far along the chromosome does the co-expression of the non-coding RNAs and the messenger and the coding RNAs, how far does it extend? I mean, you said it has to be immediately adjacent, but does it go kilobases, megabase pairs, what?
I never said immediate. I said next to. Yeah. And in some cases, next to could be 100 kb away.
I was wondering, are these promoters of the non-coding RNA, are these localized at sites that you would associate with enhancers? And is there any relationship between what you're calling the pcRNA and an enhancer RNA, or what the relationship might be?
Yeah. So at the moment, we're doing the analysis of modifications that would tell us which ones of these promoters are actually enhancers. We know that very few of them are already mapping at enhancers, but others definitely are not. So there may be a combination. But we don't know the result yet.
Hi there. I was wondering, all the targets of the, or most of the genes you described as having pcRNAs are also polycomb targets. And so are they similar to or equivalent to the polycomb inhibiting non-coding RNAs that Leonie Ringrose has also described?
As I said, we're not looking at anything individually because we have too many. We haven't looked at that. It's an important question, and we will address it, but it's not, at the moment, it's not material to our overall goal, which is to find the generic function of these things. And I would say that some of these will be in there. I just, we haven't looked.
Tony, it makes me really excited and maybe to move into non-coding RNA. It's really fantastic to listen to your talk. The pcRNAs target upstream, downstream, middle, it could be anywhere. And that's kind of unusual. And I wonder if any other class of RNA does that, and whether it would distinguish the function of what it does, whether epigenetic modification comes in again, as you have already shown for the one class you did. Could that be the one that distinguishes where to target?
Well, we would like to hope that, because then it would give us specificity for targeting a specific RNA. That would be our plan, to find that out. At the moment, we don't know the discrimination between the different subclasses. At the moment, we can only class them in terms of position. So that is the only definition we have at the moment. In terms of function, we don't really know how they discriminate from each other.
So from the data set that people have done that work on modified RNA and did pull-down experiments, if you go into these data sets, do you find pcRNAs in there?
I don't think we've done, no, we haven't done analysis, but there will be a lot of your favorite and a lot of your favorite non-coding RNAs will be in there, okay?
But they will be rare,
but some
right?
of them
They
won't.
only target few genes, you said, so it should be significant if you pull them out.
In your selection of pcRNAs, you mentioned that they have to be spliced. Is there any reason why they have to be spliced, or?
Well, that helps us to define exactly where we don't want it to overlap, the RNA to overlap with anything with the coding. That allows us to stay away from interference of the coding, and therefore we chose to only use splice.
Thank you again, Tony, for asking.